Hello, and welcome back to Introduction to Genetics and Evolution. This is one of my favorite parts of the course. This is what people typically think of when they think of basic genetics, and that is basic single gene inheritance. In the last video that you saw, we talked about the process of mitosis, which starts with a diploid cell having two copies of all genetic information and forming two new diploid cells that are genetically identical to each other and genetically identical to the parent cell from which they came. Now, meiosis, which is what we'll be focusing on here, you start with a diploid cell again, having two copies of genetic information, and you make a haploid gamete. It has only half the genetic information. Now, this haploid gamete comes together with another haploid gamete. Boom! Fertilization happens, and you have a new diploid cell that is different from either of the two parents. That is really cool. So let's talk about that briefly. Meiosis and fertilization are needed for what's referred to as Mendelian inheritance. Gregor Mendel was a monk who actually described this process very elegantly and identified how this actually worked. He lived from 1822 to 1884 in Austria. What he worked with were pea plants. He identified a number of traits that he was studying with these. So as you can see here, seed form being round versus wrinkled, seed color being yellow versus green, pod form inflated versus restricted, etc., etc. There are a lot of traits he looked at. What he found is that some forms he referred to as, quote, dominant. Now, dominant meant that if you bring together a round form and a wrinkled form and cross them together, the offspring would actually just be round. It is not intermediate, but would actually just be round. So from this, he identified some simple rules of inheritance. Now we can understand dominance and recessivity from these simple forms. So let's use, in this example, true breeding green pods and true breeding yellow pods and cross them together. Okay, now when I say true breeding, I mean all of their offspring are when they're bred with others that look like them, look exactly the same. Now, what we'll do in this case is we'll identify the genetic factors that they have, or the alleles that they have, using a letter. Typically, but not always, the capital letter is associated with the dominant form. So in this case, the true breeding green pods are dominant. So it is big Y, big Y. Again, it has one copy that came from its dad, one copy that came from its mom. What gametes can it give? Well, it's only got big Y, so its gamete would have to be big Y with respect to the form of this pod, with respect to it being green. So these guys right here, the yellow pods, they are what's referred to as recessive, and I'll show you why that is in just a second. They are little y. Again, they have, little y that came from dad, little y that came from mom. All they have to give are little y's. The F1s, or the offspring of these two things, the F1 is as offspring from a cross. Well, they'll get the big Y from this person, or from this plant, and the little y from this plant. So they are big Y, little y. Nonetheless, they are all green. So in this case, we have a masking of the yellow color by the green copy, right? The masking of the yellow color by the green copy. So big Y, the green one, is definitely dominant. Little y is definitely recessive because you don't see it when you have one of each in there. Now what will this plant over here give off? Well, he has two different gene copies. He has a big Y and a little y. So he could give either of these to his kids. So what happens when we breed these F1s? So this big Y, little y are referred to as heterozygous. Heterozygous meaning having two different alleles at a particular gene that we're looking at, in this case being the pod color gene. Right? So they have both, they're heterozygous. So we can use what's referred to as a Punnett square to follow patterns of inheritance. Now, it's equally likely for it to give one copy as the other copy. So we'll take a boy that's big Y, little y. So here's your boy, big Y, little y. Here's your girl, big Y, little y. Now, half of the gametes from the boy will be big Y. Half the gametes from that same boy will be little y. Half the gametes from the girl will be big Y. Half the gametes will be little y from the girl. So the possible combinations, the boy might give this big Y, the girl might give this big Y, and you may get a big Y individual. Well, we know that is green. The boy may give big Y, and the girl may give little y. Well, we know when you're heterozygous, you're still green. The boy may give little y, and the girl may give big Y, still green. And only in this case, when the boy gives little y and the girl gives little y, do we get a yellow pod plant from this? So the ratio we should get is 3 green to 1 yellow. And that, in fact, is actually the ratio that Mendel saw. So here's some actual numbers from Mendel. He saw 428 green and 152 yellow peas from this cross. That's very similar to the expected numbers. So that's great. Now, Mendel put out various laws, and these laws have postulates within them. So let's go over that briefly. So Mendel's first law has 3 postulates. First, that we have unit factors in pairs. So that basically every gene, you have 2 copies of it. This is assuming you are a diploid. So you get 1 allele from your mom, and you get 1 allele from your dad. Similarly, if you are a boy like me, you will give 1 allele to your kid. Your spouse will give 1 allele to the same kid as well. There's also this feature of dominance or recessivity. You don't always see this, and it is possible sometimes to have, say, a red plant and a white plant come together and make a pink plant. But sometimes you do see this, where one form completely masks how the other form may look. So you can have a green and a yellow pod as you saw, and the offspring looks just as green as the dad. And finally, we have the equal segregation of gametes. Right? So that you have these 2 factors, the big Y and little y, and you're equally likely to transmit either the big Y or the little y to one of your kids. You could transmit the big Y sometimes, and the little y sometimes. You don't have any control on it, but it's a 50-50 shot. So let me give you a problem to try. First, I'll do one with you, and then I'll give you one just to try on your own. Let's imagine you're a farmer working on corn. Okay? And let's say you have pure breeding tall and short strains of corn, and you've heard that this is caused by a single gene. This, by the way, is very, very unlikely, but let's just pretend that's the case. So you cross the tall and short strains together, and in this case you get strains that are intermediate in height. So in this example, we don't actually have dominance like in the previous example. So you cross these intermediate height corns together, what would you see? Well, let's work through this together. So again, we started with pure breeding tall and short strain. Let's imagine the alleles we're working with are big T for tall and little t for short. Okay? So let's imagine these are the 2 forms we're working with. Now, what would these intermediate strains be? Well, since we said that we're crossing a pure breeding tall, which presumably was big T, to a pure breeding short, which presumably would be little t, the intermediate ones would presumably be big T, little t, big T, little t. So what are we going to get in the offspring? Well, there's a lot of possibilities. We could get big T, so this is big T coming from dad, big T coming from mom. We could get big T, little t. We could get big T, little t, and we could get little t. So in this case, this is little t from dad, big T from mom, or in this case, this is little t from dad, little t from mom, etc. So what do we see with this? Well, in this case, we have 1/4 here would be big T, which we've identified that as being tall. Big T, little t, we've identified that as being intermediate, so it would be 1/4 intermediate. Okay. Down here we also have big T, little t, so 1/4 intermediate. And then finally, 1/4 short. So basically, 1/4 would be tall, half would be intermediate, and 1/4 would be short. So the ratio would be 1 to 2 to 1. Okay? Let me give you an example now to try yourselves. So this is starting similarly. You're a farmer working on corn. You have your pure breeding tall and short strains together. You cross the tall and short strains together and you get intermediate height corn. This is again very similar to the previous problem. You cross the intermediate height corn to the tall corn now. So you're crossing intermediate height corn to pure breeding tall corn. What will you find? Welcome back. I hope that was not a particularly difficult problem, but let's work through it nonetheless. Okay? So, you started off, in this case, you had your intermediate corn. So this is just like the previous example. However, rather than breeding the 2 intermediates together, you're breeding intermediate corn with pure breeding tall corn. So this is a little bit different than last time. So here's your pure breeding tall corn. We'll just say that's the female. It doesn't really matter which one's which. And we'll say your intermediate was the male down here. Well, in this case, there's actually fewer possible offspring. So you can get big T from here, and you get big T. Here you get big T from here, big T from there, big T again. So you can see half the individuals are big T. You can have big T from mom and little t from dad. And again, big T from mom and little t from dad. So in this case, unlike in the previous example, you would get 1/2 tall corn, and you would get 1/2 or 2/4 intermediate sized corn. You would not, in this case, see any short corn coming out from it. Now, I hope this illustrates something to you, especially contrasting to the P example. And what I hope this illustrates is that dominance does not matter in how offspring will... dominance matters in how offspring will look, but it does not matter in how gametes will pair. You know, we can use the same kind of Punnett square, for things which exhibit dominance as things which do not exhibit dominance. Because dominance only affects how things look. It does not affect how pairing happens. Okay? So let's try out a medical example in this case. So about 12% of women on average get breast cancer. And there are known mutations in the FGFR2 gene that are associated with an increased risk of breast cancer. Okay? So let's call in this case the non-mutant form big F, and let's say that they have the default 12% risk of cancer. If you're heterozygous, if you're big F, little f, you have a 20% higher chance, so you have about a 15% risk of getting cancer. If you're little f, this is the worst one, you have a 19% risk of getting cancer. Okay? Now, in this fictitious example, let's say that you're going out someplace, you're an attractive female, you meet somebody, sitting in a cafe, you have a nice chat with them, and let's pretend this is in the future. Let's say it's 10 years from now. They leave. You pull out your little iPhone, and you go, boop, and you use the DNA scanner on their coffee cup, and you say, oh my goodness, he is heterozygous for the FGFR mutation. So let's assume that you are homozygous big F, so you have the lowest probability of breast cancer, but your potential hubby is a heterozygote. He is big F, little f. And again, I put here all the probabilities so you can see what everything was. What is the probability that your daughters would get breast cancer if you were to have kids with this potential hubby? Basically, how much does it increase it relative to if you had kids with somebody who was just like you? Well, we can do this very easily. So your potential hubby, we say, is big F, little f. You are big F. Okay? So we're just looking at your daughters. We're not worrying at all about your sons in this case. Well, half your daughters will be big F. So half your daughters will be just the same as you. Half your daughters will have this mutation because they'll have the thing coming from your new hubby. So there's a slight increase, but these ones have a 12% chance of getting breast cancer based on this gene alone. Of course, we're not considering everything else like lifestyle and other genes. These ones have a 15% chance of getting breast cancer. So on average, instead of having a 12% chance of getting breast cancer, your daughters would have about a 13.5% chance of getting breast cancer. That's averaging all four of those numbers together. So really, the guy is only increasing your odds of having daughters with breast cancer by about 1.5%. So you can decide if that's important enough or not in terms of whether you want to keep this hubby. And we'll see if this sort of iPhone app ever comes about in the future. That's just a fictitious example and a humorous one just to give you an idea of what's happening here. Now, we can do the same type of cross with unknowns and actually infer the parents. So albinism, being an albino, is inherited as a recessive in humans. Now, what if you have a case of a non-albino mom and an albino dad? They have 8 kids. Now, let's say 4 of these 8 kids are albino. Can you infer, just from this information on this slide, what the genotypes are of the parents? Welcome back. I hope that one wasn't too difficult for you, but let's go ahead and work this one out. So we have a non-albino mom and an albino dad. They have 8 kids, 4 of which are albino. I said albinism is inherited as a recessive in humans. Because it's recessive, since the dad is albino, the dad necessarily has to be homozygous for the recessive. So let's call it A. So dad has to be little a. Okay? Now mom is non-albino. So she could be big A, or big A, little a, right? Now, what if mom was big A? If mom was big A, and dad was little a, all the kids would be big A, little a. So none of them would actually be albino. So given that we know that 4 of the kids are albino, we can cross this out, and we can say, in fact, mom must be big A, little a. So in this case, from looking at just the phenotype, from looking at how the individuals look, that's what I mean by phenotype, and from looking at what happens with the kids, we can actually infer the genotype. Basically, what is the underlying genetic components? What alleles do they have within them, and what combinations? So, I hope this was useful, and I hope you'll enjoy it. We'll do some more complicated things later on, and you'll have some practice problems to work with as well. Thank you. Hello, and welcome back to Introduction to Genetics and Evolution. In the previous video, we talked about basic single gene inheritance, or basic Mendelian inheritance. It's called this because this was elaborated first by Austrian monk Gregor Mendel. We saw how you get single copies of genes or alleles from one parent, and then they combine with single copies of genes or alleles from another parent. In this video, we'll talk about a special case of inheritance, that of X-linked inheritance. What happens when genes are on the X chromosome that some of you may have heard of? The other thing we'll talk about is following what happens with multiple genes. This is something we'll be doing quite a bit later in the course, but we'll begin that now looking at the principle of independent assortment. This is something that was also elaborated by Austrian monk Gregor Mendel. Now, some chromosomes have slightly different patterns of inheritance from what I described previously in terms of the single copy from males, single copy from females going to offspring. And this was first hypothesized partially because there are some hereditary diseases that pop up much more frequently in males than in females. And this is true even when both sexes are likely to get the disease. You just see a lot more men having it than women having it. There are some cases of muscular dystrophy, for example, which fit this description. Now, Drosophila geneticist Thomas Hunt Morgan studied this around the turn of the 20th century. He was working with fruit flies, and specifically working with a white-eyed mutant fly. He found that inheritance was sometimes different depending on the sex of individual. That if you tracked what was happening with particular genes in males versus in females, you're getting completely different patterns. He inferred from this the principle of sex linkage, or X linkage. We now know that there are some genes that are actually not present in two copies in all organs. That quite often you'll find that in humans, males may have one copy of a gene, and females may actually have two copies. And these are genes that are on what's referred to as the X chromosome. So often across species, males are XY. So they have one copy of the X chromosome, and instead of having a second copy, they have this other chromosome called Y. Whereas females have two copies, so females are called XX. You've probably heard this before. Now, there's not the same set of genes on the X as on the Y, despite the fact these two chromosomes pair and separate. And you'll see the principle using them is very similar to what we discussed before, except that the X has one set of genes and the Y often has a different set, and in fact, often has very few functions. In humans, one of the few genes on the Y chromosome is a gene which basically makes the embryo become a male. Now again, in this case, you get different patterns of inheritance depending on who is the mom, who is the dad, and whether you're looking at male or female offspring from them. Now I mentioned muscular dystrophy. Another example is green color blindness. Now this is an X-linked recessive in humans, meaning that if you're a female and you have two copies of this particular mutation, then you will be colorblind. Males only get one because it's on the X chromosome. So if they have the one copy, they are colorblind. This picture on the screen depicts what somebody with green color blindness may actually see when you're looking at the picture. So in the picture on the left, it has the number 74 very clearly visible. In the picture on the right, if you're green colorblind, you actually won't see the 74. In fact, you may see a different number in there. Now, inheritance of genes on the X chromosome can be studied very similarly to what we described, and again, using the Punnett square, just like what we did before. And what happens in this case is you just insert a Y instead of the second allele that would go into males. So let's try this now. Let's imagine that you're studying red-green color blindness. Something that I just mentioned is something that's inherited on the X chromosome. Well, let's say hypothetically that you have a male which is red-green colorblind. So here he is. Here's his X chromosome, and I'll put a little RCB on there for red-green colorblind. For the female, let's say that she is just normal. She does not have this mutation. just has two regular X's. So we can follow what happens in this case just like what we did before. So again, the male is equally likely to either give this X, which has the mutation, or the Y. I've already drawn in the Y's there for him. So here, let's put this in. Here's the male's X, RCB, X, RCB. Okay. And the female will always give her just regular X, regular X, regular X, regular X. Now what do we see here? The male offspring are the two on the bottom because those are the ones that have the Y chromosome. You don't become a male unless you have a Y chromosome. Okay, so this is true in humans. It's not true in all species. So looking at these individuals on the bottom, would they be red-green colorblind or no? So look at it carefully. There is no red-green colorblind mutation on them, right? So the two males here would be just fine. We can write the word fine here. What about the females? Well, the females do get the red-green colorblind mutation, but I mentioned it's recessive. So they have one normal copy and one sort of broken copy. So in this case, again, the females would also be. Well, that was interesting. Well, let's try something a little bit different. Let's switch it around. This time, let's say the mom was red-green colorblind and the dad had perfect vision. Okay, so this case here is dad. He's got just a regular X. Mom's got X, the RCB mutation. X with the RCB mutation. Okay, so let's follow patterns of inheritance. Again, the dad's always giving the Y. In this case, he's giving an X. Okay. The mom is always giving an X RCB. X. So now what do we see? Well, let's look first at the girls. So the girls are the two X's. So they have one normal X chromosome and one with the mutation for red-green colorblind. So again, the daughters in this case would be fine. So this is similar to what we saw last time. However, look at the boys. The only X chromosome the boys have has this mutation. So in fact, the boys are actually red-green colorblind. So we see a different pattern of inheritance in this case, depending on whether you're looking at the daughters or the sons, and depending on who is the mom and who is the dad. Okay? So this illustrates some principles of X-linking. Now let's look a little bit more broadly in the genome. Now everything we've focused on so far has been looking at single genes in isolation. But the genome is a very big place. The human genome, for example, has on the order of 23,000 protein coding genes. These are genes that actually make protein. It has many other important things in there. There are other things that affect how much of a protein are made, et cetera. These are called regulatory regions, et cetera. Now, again, we have 23 pairs of chromosomes, one pair being that X and Y, the other pairs being ones that follow just regular Mendelian inheritance, as we described before. Now, interestingly, so you see all these 23 pairs of chromosomes in the picture on the bottom left here. They're actually all inherited independently. So let's say, for example, I get, a particular chromosome. I have a particular chromosome 2 for my dad, a particular chromosome 2 for my mom. I have a particular chromosome 3 for my dad, a particular chromosome 3 for my mom. If I give to my daughter my dad's chromosome 2, there is no way that you can know which copy of my chromosome 3 I'm going to be giving. I'm equally likely to give my dad's or my mom's chromosome 3. But basically, what's happening with one chromosome is independent of what's happening on these other chromosomes. You're not more likely to give both your dad's copies or both your mom's copies when you're looking at different chromosomes. This is referred to as Mendel's law of independent assortment. So what happens when you're studying two traits that are inherited from genes on different chromosomes? Well, let's look at two traits. In this case, I'm looking at two that show patterns similar to being single gene inheritance. They're not actually inherited through single genes. One of them is what happens when you put your hands together like this. Watch. Put your hands together like that and see which thumb is on top. In my case, my right thumb is on top. For a lot of people, the left thumb is on top. For a long time, people thought this was inherited as a single gene. That actually probably isn't true, but just pretend that's the case for right now. If you study the inheritance of this, it looks like the left thumb on top one is dominant. The right thumb on top is recessive. The other trait we'll look at is again, dealing with thumbs, because they're easy to show, is straight thumb, which sort of looks like this, or hitchhiker's thumb, which is, mine is definitely much more hitchhiker, where it comes really far back. Well, it's thought that straight thumb is dominant, so I have both recessive things in this case, whereas the hitchhiker thumb is recessive. So let's say you were following these two traits together. Let's say you're following inheritance of them. Well, let's do a hypothetical example. Let's say that you have a dad who has left thumb on top and straight thumb, having kids with a mom that has right thumb on top and hitchhiker. So let's call them, just using these letters here to make it a little bit easier to follow. Big T, big S, crossed with little t, little s. Okay. So in this case, this is referring to straight versus hitchhiker for the S, and left versus right with the T's, where capital T is a left. Okay. So let's follow what happens. Well, the offspring will obviously get one big T, one little t, one big S, one little s, right? Because there's no alternative. So here we go. Here's the kids. Big T, big S, little s. And let's say the kids marry each other and have kids. It's a little gross, I'm sorry, but let's pretend this just for a moment. What will we see in their offspring? Well, the kids can give different alleles, right? This boy right here could give his big T or he could give his little t. He could give his big S or he could give his little s. And the same thing is happening with the girl here. So it's a little bit more complicated what's going to happen. These T and S alleles are going to be inherited independently from the parents. But again, if you give the big T, you're not more or less likely to give the big S or the little s. So there, I showed here on this picture 4 possible gametes. Big T, big S, big T, little s, little t, big S, and little t, little s. Those are the 4 possible gametes you can give. Now, there's 2 ways you could try to predict what the offspring would be like. The better way, which I'll show you second, is to actually multiply probabilities. We'll come back to that in just a moment. The other thing you can do, which is maybe more intuitive but definitely more laborious, is to follow what's happening with all the gametes. Work out all 16 possibilities. So let's do that one first. I won't do the whole thing, but I'll just show you how it would go. So again, here's the thing. We can fill this in. Remember, all 4 possible gametes we have are big T, big S, big T, little s, little t, big S, little t, little s. So that's true for one of these. Let's do the other one. Big T, little s, little t, big S, t, little s. And you can follow what's going to happen with each of these things. So in this case, mom will always be giving a big T and always be giving a big S. Dad will be giving a big T and will be giving a big S. So there's one individual. Now this is going to take forever if I do this. So let me just fill in what all the answers would be. Boom. So here's all the possible outcomes there. And I have here up in the upper left corner a little cheat sheet so you can see what happens in terms of what they would look like or their phenotype. That if you have a big T, you got your left thumb on top. If you have a big S, then you're straight rather than hitchhiker. So let's look at the ratio here. How many individuals would be left and straight? Remember, left and straight are both dominant. Well, let's count them. This one, this one, this one, this one, this one, this one. Am I missing some? This one. T and big. Oh, there's one. There's one. So 1, 2, 3, 4, 5, 6, 7, 8, 9. So we have 9 that are dominant for both. Okay. How many would be right straight? Right thumb on top straight. So in this case, color. Right thumb on top straight would be little t but big S. So little t but big S would be this one, this one, there, those 3. Okay, so we have 3 that are dominant for one, recessive for the other. The opposite would also be 3. This would be what happened if you're left thumb on top but hitchhiker. That would be these 3 over here. I'll put X's this time. I misspoke. I'm sorry. This would be left thumb on top and hitchhiker. The other one was right thumb on top and straight. So again, 3 for when you have 1 dominant and 1 recessive. And finally, 1 that's the double recessive. So the ratio you get is 9 to 3 to 3 to 1. So again, there's 16 possible outcomes here. And this is the ratio we expected. That was laborious. So let's see how we could do this a little bit more straightforwardly. Well, there's a very simple thing you can use from basic probability. That if you have 2 independent events, all you have to do is to multiply the probabilities for the joint probability. Just like if you're flipping a coin. What are the odds you flip a coin and get heads twice? Well, the odds of you flipping a coin getting heads the first time is 1/2. As we flipping it a second time and getting heads is also 1/2, so it would be 1/4. Well, we can apply this same principle to this problem. Here's all the possible outcomes when you're looking at just one gene in isolation. So we have here, this is what we'd see if we were just looking at what's happening with the T gene, the one that controls left versus right thumb on top. These are the 4 possibilities when you're looking at hitchhiker versus straight thumb. So all we have to do is just multiply the probabilities to get these different genotypes. So we say big T, big S, that would be 1/16, right? Big T, big S, little s, that would be 1/4 times 1/2. The half is a quarter, so this would be, in this case, 1/8. We can fill in this whole thing. There we go. We filled them all in. Now, if we do the same thing, let's add which ones would be left thumb on top straight. Well, we can just add them up. We can do it, let's do it by 16th, so it's a little bit clearer. So there's 1 here. This would be plus another 2, because 1/8 is 2/16, right? So this would be 3. So 1 plus 2 is 3. Plus 2 is 5. In this case, plus 4 is 9. So again, we have 9/16 have left thumb straight. So again, if you do this whole thing, it becomes 9/16, 3/16, 3/16, and the last one over here being the 1/16. We get that same overall ratio, and this time we can do it just by multiplying probability. So let me give you one to try on your own, and then I'll give you a problem at the end of it. So again, assume independent assortment between A and B genes, okay? What will the genotypes and proportions be of the offspring if you're looking at big A, big B, little b, so this is the double heterozygote again, crossed with big A, little b, little b. This is a little bit different in this case. So what I suggest to you to do is to multiply the probabilities. So look at what's happening just with the A gene, and look at what's happening just with the B gene, and multiply those probabilities together. Let me give you a moment to try this out, and then just solve this problem.